Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullah. Welcome, my dear students of grade first secondary. We continue our discussion about enzymes, the chemical reactions in the living body cells. We are talking about the last part of this lesson, the science, technology, and society. We are talking here about a topic which is talking about nanobiopharmaceuticals. Nanobiopharmaceuticals. This is one word of scientific expressions explain a new science, a new generation of science, the science of nanotechnology. Maybe you have studied it in the chemistry, the lessons of the chemistry. The nanotechnology now is a very important branch of science, which can be used in treatment of many diseases, in the industry of some products, and in many purposes. Here we have a protein. Some proteins have several vital roles inside the human body. We know proteins are very important in the body of human. Protein make the hormones of the body. Some hormones are made of protein, and some enzymes are also protein. We know that when we study enzymes, they are made of a long chain of amino acids, so they are proteins. They are considered a form of proteins. And these proteins have the ability to treat a lot of diseases and disorders inside the body has been discovered. So discovering the nanotechnology or the nanobiopharmaceuticals الجزيئات العضوية الصيدلانية الميكروبية أو الصغيرة جدا. لو حبينا نترجم هذه الكلمة هتكون الجزيئات العضوية أو البيولوجية الصيدلانية التي لها تأثير علاجي ولكنها صغيرة جدا نانو. فنتكلم عن نوع جديد من الطب أو نوع جديد من الفارما من الصيدلانية، علاجات جديدة ونوع جديد من العلاج لبعض الأمراض بيعتمد على النانو تكنولوجي أو التكنولوجيا الدقيقة جدا. Where is these biological macromolecules proteins زي الانزيمز مثلا can be produced and used to treat some diseases. And these medicines are known as biopharmaceuticals. علم ايه؟ الاحياء الصيدلانيه، الجزيئات البيولوجيه الصيدلانيه. طيب disadvantage of biopharmaceuticals. ايه مشكله البايوفارماكيوتيكالز؟ الجزيئات الصغيره دي او الجزيئات الصيدلانيه دي ايه الصعوبه بتاعتها؟ لانها صغيره جدا. It is difficult to deliver these medicines directly to the target part or cell. نجد صعوبة في توصيلها للعضو المصاب. أنا عندي مثلا مريض عنده مشكلة في الكبد أو مشكلة في الليفر، يعني a disease in the liver. أنا عايز أوصل الميديسين للليفر بس، مش عايز الميديسين يروح لكل أعضاء الجسم all body parts. أنا عايز الميديسين يوصل لمكان معين في الجسم وليكن الليفر مثلا. انا محتاج حاجه توصل لي بقى ايه الميديسين ده in the body like several medicines. انا مش عايز اوديها لكل اعضاء الجسم، هيبقى اكيد ليها disadvantages، يعني مشكله وصول العلاج للمكان الدقيق او للتارجت سيل اللي انا عايز اوصل لها. طب ايه الحل في الموضوع ده؟ فكروا العلماء في طريقه جديده way to avoid the disadvantages of biopharmaceuticals. ايه الحل بقى في الموضوع ده؟ كيف نوصل الميديسين الدقيق جدا للاماكن المستهدفه للتارجت سيلز؟ After enormous development resulted from the nanotechnology. التكنولوجيا بتاعت الجزيئات الصغيره جدا دي اكتشفوها وبداوا يطوروها. Many trials are done. محاولات كثيره اتعملت to deliver these biopharmaceuticals لتوصيل هذه الجزيئات العلاجيه او الصيدلانيه الدقيقه to the infected cell. انا هوديها للسيل اللي انا عايزها بس، للليفر مثلا او للكدني او للهارت. in the body by using هستخدم حاجه توصلها لي زي ما تكون كده ديليفري مان او ديليفري بوي. الديليفري مان او الديليفري بوي بقى بتاعي ده مين؟ قال لي نانو بارازايتس. نانو بارازايتس يعني حاجه زي ما تكون ميكروب صغير زي فيروس مثلا فيروس او بكتيريا، خليه بكتيريه او فيروسيه، هاخد الخليه دي وادخل فيها الميديسين واخليها تروح للتارجت سيل، يبقى نانو بارازايت، بارازايت صغير زي مثلا ايه الفيروس، واودي وادخل في الفيروس الميديسين اللي انا عايزه، واقول للفيروس ده روح للمكان اللي انا عايزه. leads to the arising of a new field called a nanobiopharmaceutic، علم العلاج بايه؟ بالجزيئات البيولوجيه الدقيقه. And the products used in the field is called nanobiopharmaceuticals، العلاج بالايه؟ جزيئات الايه؟ الدقيقه. تمام؟ يبقى احنا بنتكلم على علم جديد في منتهى الاهميه، كيف نعالج امراض كتيره جدا بحيث نوصل الميديسين للخليه اللي انا عايزها، كاني معايا ديليفري مان. ديليفري مان ده عباره عن ايه؟ نانو بارازايت. نانو بارازايت، النانو بارازايت دوت ايه؟ هيوصل لي الميديسين للتارجت سيل مباشره directly without need for the medicine to go to other cells, only to the target cell. Okay. Okay. Now this is the end of the lesson of enzymes, and we are gonna to solve some questions. We start solving in the last session, and we will continue today solving, inshallah, more questions. But in the beginning, I have some questions from the knowledge bank. I have some questions from the knowledge bank. I want you to watch with me. Okay. We'll see together now some questions from the knowledge bank sent by Ni'ma. Thanks, Ni'ma, for these questions. We'll solve it now, inshallah, together. The first question here we have, choose starch is formed from many repeats of these carbohydrates. Which of them? Alpha glucose, fructose, lactose, or beta glucose? What is the correct answer? I said before that the knowledge bank has many questions from outside our curriculum, from outside our lessons. We didn't study, okay? But we have to solve. We will solve it for you to make you more confident, only to make you more confident. But it is from outside what we have studied. We didn't study alpha glucose or beta glucose. But the answer here, the starch is made of alpha glucose. The starch is made of alpha glucose. This is the correct answer. Okay. The second question here, the second question, he says, the following is a list of carbohydrates: alpha glucose, beta glucose, lactose, cellulose, starch, glycogen. We have studied lactose, cellulose, starch, and glycogen, but we didn't study alpha glucose and beta glucose. No problem. No problem. We will answer the question. We will answer the question. No problem at all. To be confident. Only to feel you confident, more confident. Okay? For the following statement, state the carbohydrates from the list that is being described. ماشي. يبقى احنا هنتكلم عن ايه دلوقتي؟ These carbohydrates is primary component of the plant cell. Primary component of the plant cell wall. The cell wall is different from starch. Starch is made of alpha glucose, but the cell wall is made of yes, beta glucose. It is made of beta glucose. This is also we haven't studied, but you have to know the cellulose is made, the cell wall is made of cellulose, and cellulose is made of beta glucose. The cell wall is made of beta glucose. The answer here will be cellulose. The answer here will be cellulose. This carbohydrate is primary component in the plant cell wall, though it is beta glucose. The cellulose is the cell wall, but it is made of beta glucose. The cellulose is The cell wall is made of cellulose, but it is the primary component of cellulose is what? Is beta glucose. Okay? So the primary component of the cell wall is a beta glucose, but it is made of cellulose. The cellulose made up of a beta glucose. The type of glucose which bend together to make cellulose is called beta glucose. Okay? And we haven't studied, yes, I know we haven't studied, but for you to be confident only, I think this question cannot come in the exam because we didn't study in our curriculum. We didn't study in our curriculum. So the answer here is beta glucose. Okay, let's move to another question. These carbohydrates act as sugar storage component in the animal cell. Sugar storage component in the animal cell. Okay, which of them is a sugar storage component in the animal cell? Glycogen or alpha glucose or beta glucose or starch? We remember the animal cell has the glucose is stored as a form of a glycogen. Very good. The answer is glycogen. The animal, it is called the animal sugar. The animal storage sugar, which is a glycogen. So the correct answer here is a glycogen. Next question. Glucose can be stored as a polysaccharide, such as starch. Glucose can be stored as polysaccharide starch in the plant cell. Okay, in the plant cell, the glucose is stored in the form of starch, but in the animal cell, we said it is stored in glycogen. Okay, what is the polysaccharide? Act as the primary unit of glucose storage in the animal cell. Yes. Here we are talking about the primary, act as primary unit of glucose storage in the animal cell. Yes. Is it chitin or alpha glucose or glycogen or cellulose or glucagon or cellulose or glycogen? Again, the glucose can be stored in polysaccharide starch in the plant cell. Okay, the plant cell, it is a starch, but in the animal cell, it is what? Yes, in the animal cell, it is glycogen. In the animal cell, it is a glycogen. The correct answer here is a glycogen. Okay, the answer is glycogen. So, chitin not correct, alpha glucose not correct, glucagon, this is a hormone. Okay, and cellulose, this is the cell wall, and the answer is, the correct answer is a glycogen. Is it okay? Okay. Now move to another question. We have here question number 7. Which of the following is not an example of polysaccharide? Which of them is not considered as a polysaccharide? Okay. Starch, cellulose, glycogen, or galactose? Of course, it is galactose. Of course, starch is polysaccharide. It is complex carbohydrates or complex sugar. Cellulose also complex sugar. Glycogen also is complex sugar, but only galactose is monosaccharide. So it is not a polysaccharide. The correct answer is a galactose. Okay, the milk sugar, the found in the milk sugar. Okay. Okay. Now move to question number 8. Give the molecular formula of the molecule that is removed when two monosaccharides are joined together. Which is removed when two monosaccharides join together? You remember when we join glucose with glucose or glucose with fructose or something like this from monosaccharides, we make disaccharide. We remove a molecule of a horse. We remove a molecule of water. Yes, excellent. H2O. And this process is called a polymerization, or it can be called also condensation. By removing a water molecule, we make polymerization. We join two molecules of monosaccharide to make one disaccharide. Okay, one molecule of disaccharide. Now move to another question. Which of the following is not primary function of carbohydrates in the human body? Again, which of the following is not primary function of carbohydrates in the human body? Not function of carbohydrates. Number one, larger complex carbohydrates can be broken into fatty acids in hormones, in making hormones. What is the relation between carbohydrates and fatty acids? No relation between carbohydrates and fatty acids. Of course, this is the answer. This is the answer. But we will read the other answers. Carbohydrates act as storage molecules in to help regulate the blood glucose concentration. Or simple carbohydrates can be used to build larger molecules. More complex carbohydrates that can be used as a cell components. Okay, carbohydrates provide substrates that can be broken down into cellular respiration to release energy. Of course, the correct answer is number one. No relation between carbohydrates and fatty acids. If carbohydrates are broken, they will give glucose. If the glucose is burned, it will give energy. But there is no relation between carbohydrates and fatty acids. Okay. Now move to the next question. Which of the following is an example of monosaccharide? Lactose, saccharose, glucose, and maltose. Which of them is monosaccharide? It is so easy. Yes, it is so easy. It is glucose. Very good. Glucose is monosaccharide. But lactose is disaccharide, saccharose is disaccharide, maltose also is disaccharide. And you remember lactose is made of one molecule of glucose and one molecule of galactose. And saccharose is made of one molecule of glucose and one molecule of fructose. And maltose consists of two molecules of a glucose joined together. Okay. And remember, the bond is called glycosidic bond. Glycosidic bond. And the way of condensation and the way of bending is called condensation or a polymerization. Okay. Let's move to another question. This question here, number one. Question here, number one. What is the type of reaction occurs between two or more monosaccharides joined together? Don't look at the answers made here. Maybe this answer is wrong. Okay. Ne'ma, maybe you make some mistakes in this answer. We'll correct them now, inshallah. Now let's read the question again. Let's read the question again. Okay. What is the type of reaction occurs between two or more monosaccharides joined together? What is the type of the reaction? Is it reduction? No, of course. Substitution? No. Condensation? Very good. It is correct. Hydrolysis? No. Oxidation? No. The answer is A. Yes, condensation. Condensation. The correct answer is what? Condensation. When two monosaccharides join together and they condense, making a disaccharide. This answer is A. Condensation. Of course, not hydrolysis, not oxidation. Okay. Let's move to another question. What is the type of the bond formed between two monosaccharides when they join together to form a disaccharide? What is the name of the bond? Is it the peptide bond or phosphodiester bond or glycosidic bond or ester bond or hydrogen bond? Yes, very good. The correct answer is A, glycosidic bond. Glycosidic bond. And the way of combining is called the condensation. Remember. Okay. This bond between two monosaccharides to form a disaccharide is called a glycosidic bond. Let's move to another question. Which of the following best describe the relationship between monosaccharide and polysaccharide? The relationship between monosaccharide and a disaccharide. Okay. Number one, monosaccharides can be broken to form a polysaccharide? Of course not. Of course, not. So monosaccharides can be joined together by a bond called polysaccharide, by a bond called polysaccharide. Polysaccharide is a bond called polysaccharide? No, of course, this is not correct. The polysaccharide have complementary bending sites to monosaccharide? Of course not. Of course, this is wrong, also wrong. And this is wrong. And this is wrong. So the correct answer, I think it is number D, letter D. Many monosaccharides can join together to form a polysaccharide. This is the correct answer. Very good. This is the correct answer. Many monosaccharides can join together to form a polysaccharide. This is the correct answer. Okay. Let's move to another question. The following is a list of carbohydrates: alpha glucose, beta glucose, and remember, alpha glucose found in starch, beta glucose found in cellulose, lactose, which is monosaccharide, disaccharide, fructose, which is monosaccharide, saccharose, which is disaccharide. Remember these molecules, okay? Remember them very well. The first question here we have, for the following statement, state the carbohydrates that from this list is being described. Cellulose is formed from many repeats of these carbohydrates. Cellulose, yeah. Cellulose is made of a beta saccharide, beta glucose. Very good. The correct answer is beta glucose. Cellulose is made of beta glucose, and we didn't study. I know we didn't study, but I solved it to you to feel more confident. I think it cannot be in the exam. Not all the questions in the knowledge bank can come in the exam, because some question we haven't studied in our curriculum. But to feel more confident only. Now we have done all questions, and we can go to Al-Mu'asir book. Okay, we can go to Al-Mu'asir book now to solve some questions in Al-Mu'asir. Okay, maybe we solved some questions, and now we will continue some questions from Al-Mu'asir. Now we reach question number 7. Question number 7. Here we have the opposite graph shows the effect of the enzyme on the activation energy of the chemical reaction. We know the enzyme decrease the activation energy. We know that the function of the enzyme to decrease the activation energy needed to start a certain chemical reaction. We know that the enzyme reduce or decrease the activation energy needed to start a certain chemical reaction. Okay, the arrow that illustrates the reduction of the activation energy due to the addition of the enzyme is, okay, we add enzyme, okay, and the activation energy be reduced, the activation energy become reduced in the point X or Z, in the point X or Z, okay, or Y, or what point? Now I think it is clear that Z is the correct answer. The reduction of activation energy at point Z, it is the correct answer. Okay, it is clear that the enzyme reduces the activation energy. If the activation energy in the without the enzyme was Y, and it will be with the enzyme, it will be less, and it will be Z. Okay. Now move to another question. Question number 8. The following figure represents a chemical reaction. You can see here two components by the enzymes. They join together to make one component. Two substances join together to make one substance. Okay. So, which of the following figure represents the enzyme? We have to focus on the activation site. The activation site, the activation site of the enzyme must be suitable for both of the 2A units. The enzyme must receive the 2 units. The enzyme must receive the 2 units, this unit and this unit. So, which figure A or B or C or D can receive the 2 units? Yeah, the enzyme maybe you can here you can remove this answer and this answer. We cannot choose them, okay? Because they are not suitable for the 2 units. Maybe they are suitable for 1 unit only. Now we have A and B. Which of them suitable to put the 2 units in them? The activation site may be suitable for both of them. I think this one suitable for one only, and this A suitable for this figure only, and this shape only. But this one is correct, I think, because it is suitable for both the 2 units can join here. The first one can join here and can enter here, and the second substance can enter here. So I think the correct answer will be letter B. This is the correct answer. Is it okay? So the site, it is called activation site, or the active site of the enzyme can receive the 2 substrate, can receive the substrate. Okay. Let's move to another question. Let's move to another question. Which of the following graphs shows the concentration of the substrate on adding the enzyme to it? The concentration of the substrate on adding the enzyme to it. Okay. This one here, the concentration of the substrate increases by adding the enzyme. And this one, the concentration increases and then decreases. And this one is the concentration of the substrate decreases. And this one, the concentration of the substrate still as it is. Which is correct? I think it is clear when we add the enzyme, the substrate changes. Decrease, the amount of substrate changes. So I think the correct answer will be C, because the substrate start changing. After adding the enzyme, the substrate start changing. The substrate start changing to another product. So the substrate concentration decreases. Okay, let's move to another question. In the enzymatic reaction in stomach, the product's concentration increases. Increases by what? The product of the concentration increases. How to increase the product? By increasing the concentration of inhibitors. بنزود كمية المواد المثبطة. Of course not. By increasing the concentration of hydrogen ions. Of course not. The enzyme will not work properly. By increasing the substrate, it may be correct. Let's read the last one. When the concentration of the enzyme decreases at the normal body temperature, of course not. So the correct answer will be what? The correct answer will be C. The result will increase if there is increase in the amount of substrate. By increasing the amount of substrate, there is increasing in the concentration of the product. Is it okay? Let's move to another question. Study the following figures, then answer. We have S1, S2, S3. They are 3 different enzymes for substrate, different substrate. Okay, diagrams for substrate, substrate 1, substrate 2, substrate 3, and enzyme 1, enzyme 2, enzyme A3. Diagram of the 3 enzymes, enzyme 1, enzyme 2, and enzyme A3. Okay, now what is the question? Which of the following statement is correct about the enzymes that are present in the diagram? طيب نشوف كده diagram تاني. I think enzyme E suitable for what? For substrate 2. And the enzyme E2 suitable for S2 and S3. And the enzyme E3 suitable for S1, S2, and S3. It has an active site, 3 active sites, so it is suitable for 3A substrate. E1 suitable for only S2. E2 suitable for S2 and S3. E3 suitable for the 3 of A, the substrates. Let's read now. The enzyme number 1 is less specific, أقل تخصصا than enzyme number 3. نشوف كده تاني. enzyme 1 less specific، لا ده 1 ده very specific، ده 1 شغال مع S2 بس، يبقى ده very specific، يبقى 1 دي wrong، 1 دي ما تنفعش لأن enzyme 1 ده شغال مع مادة واحدة بس، يبقى ده very specific. طب enzyme number 1 is more specific than enzyme number 3. This may be correct. This may be correct. Is more specific than 3. This may be correct, but we have to read the others. This may be correct. Enzyme 1 is more specific because it is specific for 1A substrate only. Enzyme number 2 is less specific than enzyme number 3. 2 work with a 2 substrates. 3 work with 3 substrates. So 2 is more specific. So this answer is wrong. This is a wrong. This will be wrong. Okay. All the enzymes are highly specific. لا طبعا. يبقى the correct answer letter B. Enzyme A is more specific. It can react with one substrate only. Okay. Let's move to another question. If you know that amylase enzyme help in starch digestion in the mouth, therefore the activation energy needed to hydrolyze starch on adding the enzyme. The activation energy need to hydrolyze starch, to give starch water, hydrolyze starch to give it water. To add water to starch. This is the meaning of hydrolyze. Okay? So the activation energy will increase or decrease, or not affected, or decrease, or vanishes. The activation energy by adding the enzyme will be, of course, we know the function of the enzyme. The enzyme decreases the activation energy. The enzyme decreases the activation energy. Okay? Okay. Let's move to the next question. On trying to carry out the enzymatic reaction in the lab without using the enzyme required for the reaction. Therefore, the reaction will not occur due to the absence of the enzyme. We say that on trying to carry out the enzymatic reaction in the lab without using the enzyme required for the reaction will not occur. No, of course it is wrong. The reaction will occur because there is no enzyme required. Occur and give different results? Maybe. occur and take much more time, this may be stronger answer. Without the using the enzyme, the reaction will occur and it take longer time. Very good. I think this is the correct answer. Without the enzyme, the reaction will occur, but it take longer time. Okay, it will take longer time. This is wrong and occur without different result. No, occur with the faster time. Without the enzyme, no faster time. Of course, it is a longer time. Without the enzyme, the reaction takes longer time. Okay, let's move to another question. The following figures represent a model of a reaction for the reaction of an enzyme inside the human body. Which of the following statements represent the letter X, Y, and Z respectively? Okay. X, Z, Y, Z. Which of them doesn't change? It is Z. So Z must be the enzyme. Which of them doesn't change? It is Z. So Z must be the enzyme. Okay. So we must choose Z as the enzyme. So the last one must be the enzyme. It may be what and what? It may be only this answer. This is only the enzyme. Now you can figure that the enzyme doesn't change. X change it into Y, but Z doesn't change. So Z is the enzyme. Okay. So Z is the enzyme. X is what? X is the substrate. Y is what? Y is the product. And Z is the enzyme. We figure it easy, very easy, because Z doesn't change, so it must be the enzyme. Enzyme doesn't change after the reaction. Is it clear? Let's move to another question, the last two questions we have today. The reason for decreasing the enzyme activity in figure Y and figure X is, we have figure Y and X, temperature 37, temperature 37, and buffer solution 7.4. Okay, but here we have something, the temperature is the same and the pH is the same. So we have something different. Must be what? What decrease the activity of the enzyme? It's not the substrate concentration is wrong. No, the substrate concentration is the same. Okay, and the temperature is wrong and the pH is wrong. The correct answer must be there is an inhibitor. There is an inhibitor. Maybe something called inhibitor affect the action of the enzyme, the activity of the enzyme. Okay, and here the last question we have today, inshallah. If the thermal range for the enzyme activity was 10 to 40, the thermal range between the minimum temperature and the maximum temperature or the optimal temperature, okay? It is probably that the maximum activity of this enzyme at what? Yes, it is this number for the number 40 is the optimal. Yeah, the number 40 is the optimal. This represents the optimal. Yeah, the maximum activity of the enzyme at 40. Very good. 40 is the optimal temperature of the enzyme. And we, by reaching this question, we come to the end of the session today. Thank you, and I hope you continue solving more questions. And I wish you the best of luck, inshallah, to solve more. And send me your answer to check, inshallah. Send me your answer to check. You can solve more questions. By solving more questions, you can become better and better, inshallah. Thank you very much, and goodbye for another meeting and another session. Assalamu alaikum wa rahmatullahi wa barakatuh.